Greetings, everyone. My name is Anastasia Ryasanova, and I'm a researcher and infrastructure coordinator at Research. I'm also a researcher at Wallenberg Wood Science Center, Department of Fiber and Polymer Technology, Division of Fiber Process and Composites. And today I would like to present to you the Resource Center for Coordination of Electron Microscopy at KTH, or shortly abbreviated as RSEM from Swedish. So basically, RSEM is a new virtual center at KTH, and it has a few goals. So one of the goals of the RSEM is to increase the knowledge and competence of the KTH researchers within the field of electron microscopy, as well as to make the EM infrastructure easily accessible to the KTH staff, provide support, provide training, organize PhD courses within the EM field at KTH, as well as provide information and guidelines on how to select correct EM techniques. And other goals include creation of the network of dedicated EM specialists and dedicated EM users at KTH. As well, also the goals include the increase of the synergy between different research units at KTH and boost up of the electron microscopy related role of KTH nationally and internationally. So the board of the RSEM includes me, Anastasia, and I'm a director of the RSEM, and I'm located at the CBH school. The chairman of the RSEM is Annika Borgenstam from ITM school. As well, the board includes also Nils Nordell from the EEC school, Philip Schock from CBH school, Matthew Filden also from CBH, Peter Hestrom from ITM, and Oskar Schenberg from SCI school. The specialists from the RSEM Center are coming also from the 3 different schools. So basically from the CBH school, it's me, Philip Schock, Karsten Mim, Passi Porhonen, Caroline Ingeborg Jägerskiöld, Gunilla Hetting, and Ina Soroka. At ITM school, the specialists include Prashant Babarivatrajan, Peter Hestrom, Venli Long, and Luba Belova. And at SCI school, we have the specialists Jonas Weissenrieder, Fei Yi, and Ladislav Kalinivsky. So let's take a closer look at the infrastructure at each school. So basically at the ITM school, the RSEM infrastructure is located at the Department of Material Science and Engineering. And the research questions mostly include the analysis of the different types of steels and alloys, as well as variety of functional materials. And the facilities are located at the Hultgren laboratory. The facilities include SEM, FIPSEM, EDS, WDS, EBSD, and the possibilities for tomographical analysis. At SCI school, the facilities are located at the Department of Applied Physics, and in particular, there are a few sites. So one of the sites is Alba Nova Nanolab, which includes the FIPSEM, Schista Electron, which includes FIPSEM, EDS, EBSD, TM, and yields, and as well as ultra-fast electron microscopy, TM. The CBH school includes also several different sites. One of them is the Department of Fiber and Polymer Technology, where I'm coming from. And basically, we have the access to SEM, TEM, and EDS. As well, CBH infrastructure includes also structural biotechnology node with a cryo-TEM setup, which is also applicable for the tomographical studies, as well as surface chemistry and corrosion science and applied physical chemistry equipment, which includes SEM and EDS setups, which are mostly used for the teaching purposes. Let's discuss a little bit more in details what is electron microscopy and how it works, since we all come from the different backgrounds. So basically, we use electron microscopy to visualize the fine structure of the materials. And roughly, we can subdivide it into 2 different categories. We have the scanning electron microscopy, or briefly abbreviated as SEM, and we have transmission electron microscopy, or briefly abbreviated as TEM. When we do the analysis in the SEM, we usually work with more bulk materials, and we perform morphological studies on the micro to nano scale, and we usually collect the signal using the secondary electrons or backscatter electrons. And the resolution in this case is usually going down to the 1 nanometer scale. When we perform transmission electron microscopy, we usually work with a transmitted signal. And thanks to that, we can improve our resolution and work with the scales at the sub-angstrom level and do the studies of truly fine structure down to atomic scale. Besides the imaging and visualization in situ of the SEM and TEM, we can also perform different types of the chemical composition analysis, both quantitative and qualitative. So for example, we can perform energy dispersive X-ray spectroscopy analysis, both in situ SEM and TEM. We can also work with wavelength dispersive X-ray spectroscopy in the SEM or electron energy loss spectroscopy in situ TEM. Besides that, we can also perform the crystallographic studies, in particular diffraction in situ, TEM, for example, selected area electron diffraction, or electron backscatter diffraction in situ, SEM. With the modern SEM and TEM setups, we can also work with in situ mechanical testing. We can also work with the different temperature regimes, including both heating and cryogenic temperatures, which are very valuable, for example, for the studies of beam-sensitive materials or liquid samples. We can also perform environmental studies to do more dynamic analysis and to see the behavior of our samples under the different conditions. We can also work at the different modes, like, for example, time-resolved ultra-fast TEM. We can also perform in situ modification and even growth of the functional materials in situ dual beams. And that's usually done with the help of focused ion beam, with which we can etch away the material and do the surface modification. Or the growth of materials can be done both with the electron and ion beam, using such techniques as, for example, electron or ion beam induced deposition, and performing different types of the lithographic patterning. As well, we can also do the tomographical studies, both in situ FIB-SEM, where we work with more large-scale three-dimensional bulk analysis, or in situ TEM, where we perform the tomography more on the nanoscale. As well, we work quite much with the advanced sample preparation, including the state-of-the-art TEM lamella lift-out in situ the dual beam, which is a very high-end methodology for the sample prep for the TEM. And we can also do the dynamic imaging. So for example, we do the patterning, let's say with the ion beam, and we do the in situ observation with the electron beam, or we perform the reactions in situ the chamber of the microscope. So let me give you a couple of examples how we can try to use the SEM, TEM, and EDS techniques for the materials which are somewhat close to the field of the life science. So here I selected the examples which are coming from the materials from trees, in particular the cellulose-based materials or cellulose nanofibrils. And here on the left-hand side, you can see how we can employ, for example, SEM, in order to study the three-dimensional structure of the capsules, the CNF-based capsules. And then also, if needed, if we include the functional components inside of those capsules, like, for example, iron-based components, we can also perform the EDS and to see the distribution and the chemical composition of these particular capsules. But when we want to zoom in and to see what's actually going on inside the cell wall, or inside the fine structure of the capsule, then to some extent we're actually limited a little bit with the resolution of the SEM, and that's the time where the TEM is getting handy. So here you can see in the bottom right corner the examples of some of the TEM images of the layer by layer deposited microcapsules based on the SEM component, based on the CNF component. On the next slide, I can show you more close-ups of how the CNF fibrils would look in the TEM and how we can see the inner structure of the collapsed cell wall. And that's particular example from the synthetic plant cell fabrication. Besides imaging, as I mentioned to you before, we can also do different types of the modification of the samples. And here you can see an example of the FIB-SEM setup. So basically combined focused ion beam and scanning electron microscope. So the beauty of this instrument is that with the help of the ion beam, we can etch away the material or sputter it away, and we can open up the third dimension. And with the help of the electron beam, we can immediately image the fine structure, which is also a very unique possibility for everyone who wants to perform more three-dimensional studies and compare the bulk structure in situ in the dual beam setup. Let's say study what kind of different porosity aspects you have or distribution of the functional materials, etc. So here you can see an example of the 3D reconstruction of hydroxypropyl cellulose. Besides that, of course, with the FIB-SEM applications, you can also do regular imaging, just cross-sectional studies, TEM lift-out preparation, and circuit editing. And another fascinating application that can be very handy for the life science applications, it's actually a cryo-SEM. And why cryo is so essential, we quite often work with the hydrated samples or even liquid samples, and we would like to see what structure they have, yeah? And simply we cannot just load wet samples or liquid samples inside the electron microscopes in many cases, because we have to work at the high vacuum. So in those cases when we don't have access to the liquid cell, we can try to plunge freeze the structure and to perform the cryo-SEM studies. So in this particular slide, you can see an example of the freeze fractured, plunge frozen, preliminary plunge frozen structure of the CNF polyacrylamide aerogels, where we were able to resolve the fine cellular structure of the foams. Going back to the TEM analysis, besides regular imaging, one can also perform the three-dimensional studies, and one can even reconstruct the individual fibrils, and that's a methodology which is also highly applicable to the field of proteins and DNA analysis. And besides three-dimensional analysis, one can also do the in situ diffraction studies to resolve the inner crystalline structure of the filaments or proteins, for example. So you've just heard a little bit of the introduction and why and how we can use the different fascinating electron microscopy techniques for our research. And now let me give you a little bit of a closer overview of the facilities which are available at the KTH. And I would like to start from the node which is located at CBH Fiber and Polymer Technology Department. Basically, you can guess already from the name of the department, Fiber and Polymer Technology. The topic of our research is related to the analysis of the beam-sensitive materials, which is not that easy because in many cases, let's say for the field of SEM, the technique is created for the analysis of well-conducting materials. So when we work with the beam-sensitive materials and non-conducting materials, we have to turn them into conducting structures. So we have to employ different grounding techniques, like, for example, the coating. But at the same time, we must ensure that we don't hinder the fine structure of our samples with the coated material. So also we should be very careful with the selection of the imaging conditions as voltage and current, because we don't want to destroy our samples. So basically at the site of, at the Fiber and Polymer Technology Department site, we work quite intensively with the development of the imaging possibilities for the beam sensitive materials. And we have field emission SEM from Hitachi, which we use for the high-end analysis, and that's also an instrument which is equipped with EDS detector from Oxford Instruments and also in situ mechanical tester. Besides that, we also have the tabletop SEM, which we use for the pre-studies and also for the teaching purposes, for example, for our master students. We also have Hitachi TEM, and that's a low voltage TEM. low dose TM, which is very user-friendly for the studies of the beam-sensitive materials as well. For the sample preparation, we use the RMC ultramicrotome when we need to cut the thin slices from the bulk samples and perform the analysis in the TEM. Moving on to another site at the KTH school. So we actually have two sites. One of the sites is at the surface chemistry and corrosion science, and another one is at the applied physical chemistry. And both of those sites, they have the SEMs, which are also equipped with EDS detectors, one of them from FEI, another one from GEOL. And these are the instruments which are quite much used for the analysis of the fine structure of the materials and also the bulk structure of the materials. The equipment at applied physical chemistry is also heavily used for the teaching different courses at KTH school. And here you can also see the names of the contact persons at each of the sites. So it's Gunilla Herting for the surface and corrosion chemistry, and Inna Soroka at the applied physical chemistry. Going to the ITM school, Material Science and Engineering Department, in particular Hultgren Laboratory. So at this site, we have two different types of the SEMs. They have slightly different profiles. So basically at one of the SEMs, one can perform both the EDS and WDS analysis. And at the other one, the GEOL SEM, one can also perform the EBSD crystallographic studies. And then the last but not least, it's also a dual beam setup, FEI Nova 600 Nano Lab, which is FIB-SEM equipped with the EDS and a BSD. So earlier today, I showed you a few more examples from that particular setup, how it can be employed. And the contact persons for this site are Peter Heström and Prashad Baburevat. Moving to the SCI school, and let me start from the nanocharacterization electron laboratory. And this facility is equipped with three types of the instruments. So it has both GLTM for the high resolution studies, including both STEM, EDS, and yields capabilities. It also has the high resolution size SEM equipped also with EDS and EBSD. And the dual beam FIB-SEM from FEI Quanta 3D FAG. So if you have any questions about this particular site, please feel free to talk to Fei Yi. The next site available through the SCI is Albanova Nano Lab, and in particular it has a dual beam FEI Nova 200. And it's Vladislav Karenivsky who is in charge of this instrument. And also at the SCI school, we have a state-of-the-art equipment, which is actually ultra-fast TEM, GEOL GEM 2100. And that's one of the most unique equipment that we have at the KTH electron microscopy infrastructure, because it's a custom built setup and it has the true time result resolution. So please feel free to talk to Jonas Weissenrieder if you have any questions about this instrument. The last but not least instrument in the RSM infrastructure is actually the cryo-TEM located at CBH Flemingsberg. And that's a TM from GEOL 2100F. And I guess that's actually the instrument that quite many of you working in the field of life science have already used, at least to some extent, or heard about, because basically for the life science analysis, the cryo-TEM is one of the most essential tools, especially if you want to work with the biological samples. And this site is also equipped with the plunge freezer, the Vitrobot from Thermo Fisher Scientific or former FEI company. And that's a setup which allows us to perform the plunge freezing of our samples and prepare the samples in the artifacts-free manner without the crystals that would presumably rupture the structure of the biological matter. And here below you can see a list of the experts and it's at this site. So Pasi Perhonen, Philip Schock, Karsten Wiemen, Caroline Ingeborg Jägerskiöld. So through the RSEM, we also have a close collaboration with the SEM format, Center of Electron Microscopy for Material Science Analysis. And the beauty of this center that it basically has a strong interaction between the Stockholm University, Uppsala University, Sverim, and KTH. So please check out the website if you have any questions. Then also at KTH, we have also another infrastructure, which is called TreeSearch. And at TreeSearch research infrastructure, we also have a strong initiative in the field of electron microscopy. And basically within TreeSearch, we work mostly with the materials from trees. So here you can see a little map which unites the facilities and the experts throughout Sweden. And these are the experts who are working with state-of-the-art high-end methodology development for the electron microscopy analysis. Not only limited to the field of materials from trees, but just under the TreeSearch umbrella, I'm telling you about the example for materials from trees. But otherwise, these are unique facilities which work with a broad range of the materials characterization pretty much for all the fields. So for example, for the cryogenic assessment, the USEM facility up in Umeå is one of the leading specialists. For the environmental TEM is basically a facility in Lund. For the environmental SEM and liquid TEM, it's Chalmers. And for the diffraction studies, it's basically Stockholm University. Advanced sample preparation, it's facilities at SLU and Mion. And then about KTH, you've heard a lot already today. And then also I would like to remind you that in Sweden, we have a few of the large centers. So the centers which are working specifically with the electron microscopy analysis. So one of them I already mentioned to you, the SEM format, but otherwise we also have the national nodes for sample preparation and microscopy, which is abbreviated as NanoSPAM, and also Atomic Resolution TEM Infrastructure Network of Sweden, or RTMI, and the National Microscopy Infrastructure. USEM belongs to the NME, basically. So some of the activities that we have through the RSEM. So first of all, we are trying to establish the center and to provide the information to all of you working at KTH about the possibilities of how and where to perform the high-end electron microscopy analysis. In particular, we focus mostly on the KTH facilities, but we also are happy to provide you more information about the other nodes available in Sweden and even internationally if needed, depending on your research question. So soon the information will be published on the KTH RSEM website, basically the information that I presented today. And that will be information about the different nodes, how to contact whom and how to apply for the support and how to presumably even get the financial support to perform your electron microscopy analysis. Besides that, we are also working with the preparation of so-called roadmap for the selection of the correct methodology for the identification what kind of electron microscopy analysis would be the most suitable for you to perform your research, depending on your research question. So here you can see a little diagram, like a flow chart, and soon there will be more information in a connection to it. So for example, where to select if you need to do the cryo-TEM or you need to do the environmental SEM, what kind of microanalysis should you do the diffraction, and etc. Beside that, under the RSEM umbrella, we are also working with the preparation of the PhD courses in the field of electron microscopy. And there will be a few courses coming up already next year, 2021. And one of the courses that I would like to give you a brief overview about is actually the course in the field of fiber and polymer technology, or basically electron microscopy analysis of fiber and polymer-based materials. And it will be the course which will include both the theoretical part and hands-on part. and it will include both SEM, TEM, EDS, and sample preparation techniques. So the theoretical part, it will be provided online. So pretty much everyone can join remotely. And for the practical part, we should be very selective this year, unfortunately, due to COVID-19 restrictions. So the practical part will be with strong limitations. And at this point, it will be mostly for the researchers from the FPT. But we are now developing the different models and different types of the courses to make it more inclusive and to make it more accessible to everyone. Because basically through the RSEM infrastructure, we would like to welcome everyone from KTH to gain more knowledge in the field of electron microscopy and to get access to the facilities and get access to the specialists. So basically it's the goal towards improvement of the synergy between the different nodes and help with competence support for the electron microscopy analysis. And with this, I would like to thank you very much for your attention. Please feel free to contact me for any questions. Here is my email, anari@kth.se. And I'm looking forward to discussing more details with you at the brainstorming session that we will have later this afternoon.